I'm looking for Prof. Collins Adeba and then Prof. Ozinel Rovizo. Are they here already? Okay. of Prof. Rasheed Adeleke. Ladies and gentlemen, kindly stand for the academic procession and the university anthem. Thank you, ladies and gentlemen. Please be seated.
Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening, everyone. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين. Just to get a bit of understanding of what was recited, this is basically the opening of the Quran Kareem, the Holy Quran, where we begin by asking Almighty Allah, by asking our Lord for help. A'udhu billahi minash shaitanir rajim. I seek refuge in Allah from the accursed devil. Bismillahir Rahmanir Rahim. I begin in the name of Allah, the most kind, the most merciful. Alhamdulillahi Rabbil Alameen. All praise be to Almighty Allah, the most kind, the most merciful, the ever merciful. Ar-Rahman Ar-Rahim, Malik Yawmiddin. And who is this Almighty? Who is this Lord of ours? Malik Yawmiddin, the Master of the Day of Judgment, the Sovereign of the Day of Recompense. Iyaka na'budu wa iyaka nasta'in. You alone do we ask for help. You alone we ask for assistance. Ihdina as-sirat al-mustaqim. Guide us along the straight path. And which is a straight path? Sirat al-ladhina an'amta alayhim. The path of those upon whom you have favored. غَيْرِ الْمَغْضُوبِ عَلَيْهِمْ وَلَا الضَّالِينَ. And not the path of those who have invoked your anger and those who have gone astray. Professor Rasheed asked me to speak something on knowledge, and with a limited time, something came to my mind. What could I speak about knowledge? Because if you ask anyone, knowledge is very vast. You can write essays, you can speak for hours. And firstly, I would thank Prof. Rasheed for granting me the opportunity to speak here on behalf of him, to come in such an audience. It's actually my first time. Before I came here, my wife told me, are you prepared to go to such an audience? I've spoken to many audiences before. Of course, it wasn't an audience like this. I've given sermons and lectures in the mosque, in our premises where the Islamic education is given. So I said, it's just one of those lectures. On this, something came to my mind on a lighter note. People suffer from different types of fears. Standing here in front is also a type of fear which I'm trying to overcome right now. So on a lighter note, there was this husband, there was a murder scene. So when the officers went to that place, he sends the sergeant and he tells them, did you arrest did you arrest the woman, the wife who killed the husband? He said, it's a very difficult scenario. He said, what do you mean? What has happened? What transpired? He said, no, the wife just mopped the floor. So the husband, when he entered the home, when he entered the house, the floor was still wet. He walked on the wet floor. So then she killed the husband. So he said, okay, we got evidence, there's cameras. He said, why are you delaying? Go arrest her. He said, I'm still scared, the floor is still wet. So on knowledge, what comes to mind? We should all try to make a prayer and ask Almighty Allah, what's that dua, what's that prayer that we learned from our teachers? Allahumma inni as'aluka ilman nafi'ah. Oh Lord, I as'aluka, ask you, ilman nafi'ah, for beneficial knowledge. One is knowledge, one is information. That knowledge which you gain, and then it is not beneficial to the community, it doesn't bring you close to Almighty, to your Lord, to your religion. It actually takes you away from the people. Or it's such knowledge which have caused interruption in your community, or because of it you have caused damage to your community. Indeed, that is not beneficial knowledge. So we should often try to make a prayer whenever we wake up early in the morning, whether we're heading towards our workplaces, towards the university, wherever it may be in different spheres of life. We always try to make this dua, this prayer. And what's the prayer we should ask? Oh Allah, grant me, oh Almighty, oh my Lord, oh God, grant me beneficial knowledge. Thank you everyone. Jazakumullahu khairan.
Assalamu alaikum. Dumelang, goeiemorgen, molweni. Thank you very much, Sheikh, for your very inspiring message. I can assure you that there's nothing to fear here. As we say in my language, all the dogs have been chained. Greetings to you all and a warm welcome to the inaugural lecture of Professor Rasheed Adeleke at the Faculty of Natural and Agricultural Sciences here in the Potchefstroom campus. I wish to acknowledge the presence of Professor Darrell Balia, the Deputy Vice Chancellor of Information Technology and Potchefstroom Campus Operations. Prof. Darrell is the main functionary today. I would like to note the presence of all other deputy deans. I can only see one currently, Professor Francois van der Westhuizen, the Deputy Dean of Research and Innovation. He will also be delivering the closing remarks. And in absentia, Professor Tebe Midupi, the Deputy Dean of Teaching and Learning, and also in absentia, Professor Lebo Katata Seru, the Deputy Dean of Community Engagement and Strategic Partnerships. Many directors are also here. I will not mention them by name, but they are also recognized. And the most important person today, Prof. Rasheed, we welcome you, your family and friends, collaborators in the industry, international partners, and the esteemed Sheikh Kari Safwan Limbada, and all staff members and visitors. And I know some of the staff members come as far as from Mahikeng and in other campuses as well. So it is quite heartwarming to see that this inaugural lecture, Prof. Rasheed, is attended very well across the campuses. Prof. Darrell, this is the 12th inaugural lecture this year. So there's 3 more that are remaining up to the end of the year. So this means that this faculty is very, very progressive, because we actually do recognize staff members that are very progressive, staff members that are well-deserving, such as Prof. Rasheed. You know, he's a very, very hard worker. You will see when I read his little resume, I'm saying little because I actually asked him to cut it down, you know. He wrote a booklet. I said, no, Prof. Rasheed, no, we don't have the, there's a lot of things that you have done, but we know. But and my task really is to introduce him. But I cannot resist, and you would know, Prof. Darrell, but you have not yet cautioned me. So until you do that, but I cannot resist not saying something about the faculty. This is just one of the faculties, that is one of the biggest faculties, you know, in the university, with the highest numbers of professors and the highest number of rated professors. The only A-rated professor in the whole university resides in this faculty. And the B-rated professors, which as from Monday, Prof. Rasheed, I will be pushing him. The B-rated professors, the largest number percentage, about 40% or so, again, comes from one faculty. So Prof. Rasheed is in really good hands. We look forward to your presentation, Prof. Rasheed, but before we go there, I would like to just read this. Prof. Adeleke was educated in Nigeria, South Africa, and Canada. He completed periods of postdoctoral programs in South Africa, the USA, Germany. For his early education, he completed his matric with distinctions in 7 subjects at the famous Olivet Heights, Oyo in Nigeria. His first degree was obtained from the University of Ibadan, Nigeria. He then proceeded to undertake his master's degree program at Rhodes University, Grahamstown, where he graduated with distinction. For his PhD program, he worked in collaborative research between the University of Pretoria, South Africa, and the University of Laval in Canada. During his PhD program, he was awarded a Commonwealth Scholarship, tenable at the University of Laval. After spending 2 years in Canada as a Commonwealth scholar, he returned to South Africa in 2009 to complete his PhD program. In 2010, he became a postdoctoral fellow at Northwest University. I must actually indicate here that I've known Prof. Rasheed long before he came here. I collaborated with him as well when I was at UNISA. So what he says here is true. Okay. And in 2010, he became a postdoctoral fellow at Northwest University. The Agriculture Research Council of South Africa appointed him as a senior researcher at the Institute for Soil, Water and Climate. While working for the ARC, established the Microbiology and Environmental Microbiology Research Group, which later became the biggest and most productive research group in the ARC. In 2018, after 7 years of service at the ARC, he accepted the position of an associate professor at Northwest University. Again, it was me who interviewed him and who accepted him. Where he established another research group in 2019 known as the Environmental Microbiology and Agricultural Biotechnology. In January 2022, he was promoted to the position of full professor by the same university. Again, it was me. Now, just backtracking a little bit, in 2013, Prof. Adeleke had the opportunity to work as a fellow of Cornell University in USA. In 2016, he was awarded another fellowship by the World Academy of Science for a cooperation visit to the Technical University of Munich in Germany. He has over 20 years experience in plant microbial ecology, specializing in plant beneficial microbes with specific focus on endophytes, such as mycorrhizal fungi, plant growth promoting microorganisms. His research theme is harnessing microbial activities for environmental protection and sustainable agricultural practices. He is presently a member of the subcommittee to the Ministerial Advisory Committee of the Genetically Modified Organisms in South Africa. In addition, he is an editorial board member, stroke associate editor for 3 international journals, the 3 Biotech, Discover Agriculture, and the Host Microbe Associations. He has published more than 110 peer-reviewed articles in international journals, and he has also a lot of book chapters that he has produced. And he has also examined MSc and PhD thesis from different local institutions and many other universities externally. I'm not going to list them all. There are just too many to list. But from various universities across the globe. And he has also examined students from other international institutions, such as the Beni Suef University of Egypt, Babcock University of Nigeria, and Botswana International University of Science and Technology. He has attended international scientific meetings and conferences in the USA, England, Germany, Scotland, and in many other Western, African, and Asian countries. He collaborates with researchers from international institutions in the world. Again, the list is there. His activities are indicative of significant accomplishments at international levels in his field of research. Evidence of his achievements is proven by his role of judging the work of others in the same or in allied fields. Such roles are clearly reflected in examples that include his appointment as a research reviewer by the National Science Foundation of the United States of America for more than 3 years, as well as a reviewer for Austrian Development Corporation. He's also a reviewer for Khalifa Universities of United Arab Emirates Furthermore, Prof. Adeleke has also acted as an external assessor for senior promotions of associate professors, full professors, and senior researchers for both national and international tertiary institutions, such as the University of Namibia, University of Zululand, and the Agriculture Research Council of South Africa. At the level of a reviewer, he has been able to serve as a peer reviewer for many international journals. Well, I suppose that I've already mentioned. He has also participated as both reviewer and panel member for the National Research Foundation of South Africa, Water Research Commission, and the Department of Science and Technology. Prof. Adeleke has also had the opportunity to serve as a session chair in international conferences, such as the Euro-Mediterranean Conference for Environmental Integration in Italy. The key achievements in Prof. Adeleke's research outputs are his innovative approaches to unique research questions. I just want to say that with those words, it is just a synopsis of the work that he has done. You know, we could spend days and days if we were to go deeper into what he has done. We really appreciate him in the faculty. We appreciate him in the university. And we welcome him as a new professor in the faculty. Thank you.
Wow. Let's unmute the microbes. Yeah, I don't know if it was what Sheikh said about the fear. But thank God Prof. Modise also said something about there is no fear here. It is who you are. So I welcome everybody once again. Prof. Balia, thank you for coming. Prof. Modise, yes, you are right. I've known you for a very long time. And the relationship has been nothing but productive. Thank you very much. Prof. Francois, I appreciate your coming for this inaugural lecture. Colleagues, I appreciate you being here. Family and friends, I welcome you once again. So, yeah, I think I'm going to break the protocol a bit by congratulating myself first. For taking this risk of putting together this diverse group of people in the same room. Family, friends, colleagues. I just have to be careful. Nothing must go wrong. It's only politicians that can take such risk. You know, they put together a lot of people in the same room and they say whatever they want to say, and people do believe them. Yeah, I'm talking of politicians. You know, today we're going to talk a lot about food chain. So there is a lot that politics and food chain have in common. I'm sure the scientists here are keen to know what I'm going to talk about, but I'm going to disappoint you. Talking of the food chain, recently we had some politicians talking about humans eating dogs, humans eating cats. That's their own idea of the food chain. That's not what I'm going to talk about here today. It's not about humans eating cats and dogs. So definitely it's not what I'm going to talk about today. So just an outline of what I'm going to be speaking about. There's a story behind me as well. I'm a human being, of course. So I will talk about myself and the research and my research group activities, and the research on endophytes in the food chain. Sorry, can I just grab a bottle of water there? So, as well as the concept of reconfiguration of the food chain that I'm going to really speak about today. Yeah. So it all started here. This is a union between my father and mother many decades ago. I won't tell you how many decades ago. Yeah, but without this union, there would not be any Rasheed. But I'm grateful for what they've done for me. So I come from a family of academics. Both my parents were teachers. So right from the beginning, if you don't study, you get yourself into trouble. So growing up, I've got 7 siblings, and I'm glad that 2 of them are here today. Mr. Adeleke and Dr. Adeleke, thank you for coming. I just want to say 1 or 2 things about one of my brothers that is here today. Yeah, at the beginning, I think it was a good role model. It helped me in many ways to establish the interest that I have in science today. So I'm saying thank you to you again. So I attended Olivet Baptist High School in Oyo, and this is one of the old pictures during my valedictory occasion. I don't know. Do you think that looks like me? I wouldn't know. I also went to University of Ibadan where we had so much fun. Yeah, trust me, it was a good time. Then there was this National Youth Service that we always did after your first degree. Well, this looks like me, isn't it? This looks like me, okay? So one of my students last week asked me something, and I decided to put this as part of the slide. He asked me that in the past, was there a certainty for me that I was going to be a microbiologist? The answer is yes and no. Why? Because, I also took some risks. I went to aviation school anyway. I spent some time in aviation, and it was also a good time. Well, having spoken about all those, there is issue of life and death. When we live, one day we will all die. And there is also that part of my history that I've lost some close family members that I will never forget. First on the list, my parents. Yeah, both of them together. It's not that they died the same day, but yes, they are no more. My brother who died last year, and a family friend, Dr. Obori. and a daughter who lived for 7 days. Okay, now, just a tribute to some people that have really contributed to my career. My mother-in-law is here, and so many of my uncles as well. Sorry, I just need to mention this again. The people that really contributed and helped to shape my career. My supervisor for the master's degree, Professor Joanna Demis. My my supervisor and co-supervisor for the PhD program, Professor Eugene Clete and Professor Damas Kassa, as well as Professor Carlos Benzidelt, who invited me here for a postdoc in 2010. So many people may not know that, but yes, we have a kind of history. I appreciate what they've done for me. He is actually the first person that made me to know so much about this campus. So let's go back to the topic for today. Research in agriculture and environmental microbiology is the theme focus that I have for my research. When I was preparing this slide, many ideas were just coming in. At a point in time, I had up to 200 slides. But Emma kept on warning me, you have 45 minutes. So I was worried that how will I be able to say everything in 45 minutes? So I tried something and it worked. Let's hope that it will work. Because we never know. So this is the research team that I have. We've done so many research in agricultural microbiology and environmental biotechnology. But I'm not going to say, I'm not going to discuss everything we've done on them today. So, but quickly, there is research that we've done on restoration and bioremediation. We've published many articles on that. We've done many research on renewable energy using agro-waste and plant microbes. We've done research on microbial diversity and plant growth promotion. And this is also related to what I'm going to be speaking about today. So this is the title of my presentation again, just to remind you. And Endophytes is going to feature very well here, as well as food chain. So please pay attention to the following keywords. It's quite important. So I will start by talking about microbial interactions, but I cannot give a lecture on microbial interaction here. I will just speak about the mutualism where endophytes are involved. You know, after COVID, people developed this kind of idea about microorganisms, that microorganisms are bad. But I can assure you, there are many good microorganisms. As a matter of fact, there could be many good microorganisms. There could be many microorganisms that are doing positive things than the bad ones. So what are the endophytes that I'm going to be speaking about today? Endophytes are mostly bacteria or fungi that live inside the tissues of plants without causing any apparent disease. So that's the definition. They live inside a plant, not on the surface of the plant. I need to acknowledge the people that I may say that first describe what endophytes are. So, but let's look at the whole concept again. There are many microbes in the environment. As we are sitting here, the air we breathe in contains microbes. The table surface, everything around us, there are microbes there. But not all these microbes can become endophytic. So how do they associate with plants? So there are two major methods that the plants acquire the microbes to become endophytic. There is vertical and horizontal transfer of the endophytes. For horizontal acquisition, you have the plants acquiring the microbes from different environmental sources, excluding the parent. For vertical transmission, acquisition is from the parent. I mean parent to the offspring. And to put this into proper context, for vertical transmission, for instance, if you take a seed of a plant, what is contained in that seed is not only plant materials. There are many microorganisms that are contained there. So those microorganisms, when you plant your seed, those microorganisms are able to move from the parent to the offsprings that are produced by the plant. So that's what we know as vertical transmission. So, but in my research group, the focus has always been on the horizontal transfer of the microbes, or horizontal transfer of the endophytes. Why? Because for the vertical transmission, it actually limits the opportunity for diversity to be improved, opportunity for the association to include many more organisms. So the focus has been on horizontal transfer of the microbes for my research group. So, and what do I mean by this? For horizontal transfer of the microbes, these microbes can be acquired from different environmental sources. It can be from the water that is used for irrigation, or it could even be rainwater. It can be from the soil. It can be from animals, such as honeybees. It can be from the air. So, but the plant is able to acquire them from different sources. How does this happen? So, when you take microbes from these different sources, we have what we call a microbial pool around the plant environment. But as I said, all of them have the potential to become endophytic, to colonize the plant, but not all of them can colonize the plant. So there is what we call filters. These filters are in three categories. There is abiotic filters, there is host-imposed filter, and there is microbe-species interaction filter. For the abiotic filters, you are talking of something like temperature and whatever, because if in the first instance, if the microbe or the bacterium or whatever is not able to survive in that environment, it can never get to the plant surface or the plant environment. So, but if these microbes are able to bypass that layer of filter, then they need to face what we call host-imposed filter. The host-imposed filter mostly is about plant chemistry, the plant immune system. The plant doesn't want anything to just colonize it. It's also very defensive sometimes. So, but some microbes will be able to bypass this as well, and then they face the other microbes that are already colonizing the plant, and they need to interact with them. So there is microbe-microbe interactions that we can also talk about. So, but our interest today is the microbes, or the interest today will be the microbes that are able to bypass all these three filters and become endophytic. So this is what I'm going to be talking about today, especially their role in the food chain. When I say their role in the food chain, what we know, the food chain starts with, just thinking if I have, okay, but that's fine. The food chain starts with the plants. The sun shines on the plants, and you have the plants growing during photosynthesis and all sorts of things. But what we don't usually see is the role that the endophytes that are associated with the plant, the role they play in that initial stage of plant growth. So these endophytes are responsible, or they are partly responsible for biogeochemical cycles such as nitrogen fixation, phosphorus solubilization, siderophore production, as well as phytohormone production, among others. Now, for you to appreciate what I'm saying very well, the air we breathe in, it contains 78% nitrogen. The question you ask yourself is, why do we then always complain in agriculture that the soil lacks nutrients? We don't have nitrogen in the soil. We want to buy chemical fertilizers. Why? Because the plants cannot utilize the oxygen that you have in the air. So you need these microbes that I'm talking about. You need them to take the nitrogen from the air and convert them to forms that are utilizable by plants, such as nitrates, ammonium, and nitrites. So this is what we've been doing. An example of this kind of association is what we have in the leguminous plant, where you have the root nodules. So we set out to ask some certain questions. One of the questions that we've asked in the past in my research is, can genetic modification of plants lead to shifts in the diversity of the endophytes and their roles in biogeochemical cycling? We all know about GM foods. And I mean, I would say two schools of thought. Some people will say GM foods are good. Some people will say, but that's not, I'm not even going to go into that space today. But I'm just going to talk about the research that we've conducted on the GM foods. So the question is, when you modify a certain plant, can that affect the roles of endophytes in that plant? So that's what we set out to do. And the first crop that we used for this experiment was maize plants. So the first thing we look at, we try to check the rhizosphere soil, which is a microbial pool for the where endophytic microbes can be recruited in the soil. So we try to check the soil and see what impact the genetic modification of the maize can actually have on them. For those who do not know about BT maize, it's a genetically modified maize plant. The modification was done to actually protect maize plants from insect infestation. So the focus here was, our experimental plan included a control plant as well, which was not genetically modified. The student that has become my colleague that worked on this is also here today. Deidre, she worked on this as well for a PhD. So if you look at this, we were able to take samples from the rhizosphere soils of genetically modified food crops as well as the non-genetically modified food crops. Here we label them, the genetically modified food crops, we label them as BT, and those that were not modified, we label them as non-BT maize plants. So for our results, it was a quick one, and we were able to see that the diversity, there is a change in the composition and the diversity of the endophytes that are associated with the rhizosphere of this soil. And that's not surprising, but we went ahead and actually checked the endophytes themselves that are associated with the plant. And we collected samples from tassels, leaves, and the stem of the maize plants, both the genetically modified plants and the non-genetically modified plants. And what we did first was to check the impact on the structure, composition, and diversity of these microorganisms. And it is clear that, we saw the differences. As we can see here, just a minute. So we can see the difference here, as we can see that the structure of the microbes that you have here, or the abundance of the microbes that you have here, the diversity as well, they are quite different from one another if you compare the BT and the non-BT maize. The BT is the genetically modified one, and the MBT is the non-genetically modified food crops. But we didn't stop there. We also checked the functional roles, how the functional roles of these microorganisms were affected or were impacted by the genetic modification. But interestingly, we saw that both the endophytes that are associated with the genetically modified food crops and those that were not modified were able to fix nitrogen. They were able to help with the phosphate solubilization, and they were also able to help with the production of indole acetic acid. So another part of this research also showed us that even the diversity that we have, the percentage of microorganisms that are isolated from the genetically modified food crops is actually more than what you have in the non-genetically modified food crops. So to answer this question, because I don't want to dwell into this too much, to answer this question, the answer is yes, but this has no negative or potentially negative, but there was no negative or potentially negative interaction that was observed or linked to the genetic modification. So we can say here that food chain may not likely be impacted by the shift in the composition, diversity, and functions of endophytes, as many of these beneficial endophytes are in good shape. So the other experiment that we conducted is to look at the rhizosphere soil again, where the endophytes can be recruited. So in this instance, we check certain, we focus on certain parameters. We check the chemical impact of chemical fertilizer application, impact of soil tillage, and impact of organic farming. For the first one, it is about the till versus no-till soils, as well as fertilizer application. So quickly, what we saw is as soon as you continue to increase the fertilizer application as a farm practice, you have changes in the diversity of the microbes that are colonizing the rhizosphere environment of the soil. And we also do what we call a co-occurrence analysis on our data. So if you look at that, the nodes that are joined here by green lines indicate positive association. The nodes that are joined by the red lines indicate, I don't know if you can see the red line clearly here, but they are there anyway. But what we observe here is the fact that there are more positive associations that are associated with no-till soil compared to till soil. So in summary, we can say that the diversity of these microbes, of these bacteria, are affected as well as the phosphate and nitrogen metabolism and the biosynthesis of phosphorus as you increase the phosphorus fertilizer application. So again, in conclusion here for this part of the study, the degree of ecological rearrangement that we have can be said to be linked to the tillage practices that reveal that the conventional agricultural practice that we have here in terms of tilling the soil reduce the complexity as well as the efficiency of the bacteria communities. So again, the take-home message here is food chain could be impacted by the fertilizer application that that some farmers practice. So the other one that we discuss here, that I will discuss here, is organic farming and non-organic farming, where we actually compare the utilization of organic manure and chemical fertilizer in certain farms in South Africa. So here, it is clear that the diversity of the microbes you have less number of microbes that are associated with the use of chemical fertilizer on the farms compared to the organic fertilizer. So though they have some microbiome that they share, but it is quite important to note that organic farming is quite better in terms of environmental sustainability here. So, and this is also reflected in the differences that we observe in the abundance of these microbes in the two farming systems that we examine. And different vegetables are also planted on the farms that we examine. So the co-occurrence analysis that we did there also gave us an indication that there are more positive interactions of these bacteria or of the microorganisms that are associated with the organic farming compared to the conventional farming where chemical fertilizers were used. So in summary, no tillage and organic farming practices have positive impact on the rhizosphere microbial communities as they provide healthy environments where endophytes could be recruited. So the take-home message here is for organic farming, if organic farming is practiced, then it could also have potential positive impact on the food chain. I will quickly speak about mycorrhizal fungi here. I've been speaking about bacteria, but here the special type of, this is a special type of fungus that colonize the roots of almost all plants. And just like most of the other endophytes, it's a give and take association. There must be there must be a mutual interest there. The mutual interest here is the plant being able to provide carbon in form of sugar to the microbes after photosynthesis. Whereas the fungus is able to mobilize nutrients from different parts of the soil and pass them on to the plant. These nutrients are typically not available for plants because of the kind of forms or chemistry that they have in the soil. But these fungi, they are able to mobilize them and make them available for the plant. So it's a very positive association. And as I said, most plants are colonized by mycorrhizal fungi. So again, we ask a question here. The question we ask here is, can the food chain be recreated in the damaged ecosystem? And this is a very interesting concept. Why? Because, yes, the food chain can be damaged, but it's easy to damage things, but is it easy to also fix them? It's not always easy to fix them. So can the food chain be recreated in the damaged ecosystem, such as abandoned mines? And the answer is provided by this study. So the idea here is the restoration of ecosystem. Can we equate it to the restoration of the food chain? It's not as direct as that, but we use some indirect indices to check for this. So what we did was to go into rehab sites in different mines where we carried out this study. And we collected samples, and we checked the plant samples that we collected. We checked them for the colonization of the root samples to check how these root samples were colonized, especially over different, over different years. So, and we discovered that, yes, these microbes are there, but we established a direct correlation between the diversity or the number, or the abundance of these microbes and the age of the rehab sites. So here, we were able to show that the arbuscular mycorrhizal fungi can actually be used as indicator of soil health in rehab sites, which can have indirect implication on the food chain as well. Because without the plants that are primary producers in the food chain, there cannot be any food chain. So if you are trying to reestablish the food chain in damaged ecosystem, it is important to have the right microbes that can associate with these plants. So again, we then move into the innovation that are out there in terms of what people have done in the past to these endophytes. So not innovation from us. So what I'm referring to is people have looked at the roles that these endophytes could play, and they were able to confirm that if they manage to screen them in different ways for these growth-promoting attributes, they can put together a synthetic community that can be utilized to actually replace chemical fertilizers. And this is another focus for my research group. But the risk in this kind of approach is if you get it wrong, you can be putting wrong microorganisms into the food chain. And I would discuss why here. So now, so we've done so many research on this, and this is the one that I'm really going to talk about in terms of checking the biofertilizer quality that we have available in South Africa. So when we check the diversity of the microbes that are contained, because biofertilizer, let me just try to define what biofertilizers are. Biofertilizers is a kind of synthetic community of microorganisms that are put together to promote plant growth. This is not chemical fertilizer, but biofertilizer is different from organic manure in the sense that it must contain living organisms. So in this case, our focus is on biofertilizers that are able to promote plant growth. So we focus on the biofertilizers that are available in South Africa, and what we discovered was the fact that, the diversity is there, but the question is, do they contain the right kind of microbes that we want? So this is the evaluation that we did, which was published as well, and it was surprising to us that most of the biofertilizers that are available, and these are fertilizers that are supposed to replace the chemical fertilizers that we are presently using in most farms. So to our surprise, it's only 3 of the 13 biofertilizer products that are, we even say, medium quality. Most of them are poor quality fertilizers. So the risk here is the fact that if you put the wrong microorganisms into the environment in form of biofertilizer, you are actually disrupting what is happening in the food chain. So now is time to talk about the concept that I was proposing. This is the direct proof for the concept that I'm talking about, to establish the actual roles of endophytes in the food chain. So we use 2 approaches. The first approach, if you go to some West African countries, you have a situation where people use leaves to wrap ready-to-eat foods. Well, we focus on that. I will tell you why. And the other one is the ready-to-eat vegetables. So at this moment, I assume that if you are sleeping, please wake up now. So at this moment, I assume that we all know that microbes are able to colonize plant leaves, different parts of the plant. But in this instance, we focus on the plant leaves. So this is, for the field work, I went to Nigeria to collect the samples, because to prove the concept, I need to look for a place where they are actually practicing this in terms of wrapping the food with leaves. So I recruited like 9 vendors, 3 per each food item: Eko, Adun, and Iru. Most of us here, we may not know this food per se because they are not common foods here. So when you say pap here, the pap that we have here, is made from corn, but the meaning of pap in West Africa is a bit different. It's also made from pap, but the texture of the food is also different. It's also made from corn, rather, but the texture of the food is quite different. But I'm not a food technologist, so I'm not going to talk about that today. So, and we collected 4 different leaf samples. One of the leaf type that they are using to wrap food is probably the one that we all know, Theobroma cacao, cocoa leaves. So we also use Ewéran, which is Tomococcus danieli. We use matchstick leaf, Astonia microphylla, as well as Ewe Budogi, Megatherium macrostachium. Professor Amo will kill me if I don't pronounce that very well. He's a botanist. Yeah, so this is the process. I'm not going to go into the details because of time of the process, but the idea here is they are preparing the food and they are wrapping them immediately with the leaves. So what we want to see here is the movement of this endophyte that I've been speaking about into the food item that people are consuming, and what could be the implication of that. So, yeah, I was there full time. One of my brothers that was here, that is here now, was also part of this because I needed many people to help with this process. So we went into the farms where they are growing the leaves to collect the samples by ourselves, and we took the leaf samples to the vendors so that we can give it to them to wrap the different food items. So this is just an indication of how the whole process unfolded. So you can see how they are putting the core in the leaves. So So, but because of time, this is an ongoing research, but what we've seen here is when we analyze the data from the leaf samples and the food samples, there is potential transfer of the endophyte from the leaf into the food. So the food we're consuming actually contains the endophytes. So you will see the clustering together here, the clustering of, just want to see if this is, okay. So you will see the clustering together. If you see this color here, so the clustering of the isolate or the microbes from the leaves and the food samples. So we still need to do more analysis on this, but there is an indication that we have movement of the endophytes that are contained in the leaf samples into the food samples. And with that said, this is what we are really proposing, that you have the plants colonized by the microorganisms, and the plant that is colonized by these microorganisms have endophytes in their leaves. And when these leaves are used to wrap the food items, human beings can consume this food. And when you consume that food indirectly, or directly now, you are consuming the endophytes as well. So, and these endophytes, when we produce waste, can always go back into the soil. That's a typical food chain that we can have in this regard. I know people like Prof. Balia, they are thinking now that, but I don't consume this kind of food, so it's fine. But I will tell you something else. So the question I'm asking all of us here, I'm a bit worried to say this part of the, to go into this part of the talk, because I wonder how many of us we want to hit after this talk. But I'm not scaring you. It's just what I have to say. So the endophytes in the food chain, how do they really get there? So for us to prove these concepts, we need a kind of idea that we show that we can consume these endophytes as they are on the plant. So if you take a plant from the farm and you take it home, if you cook the plants, you kill all the endophytes. So it's not going to work. So we cannot focus on plants that are cooked. So we focus on vegetables that are eaten raw. So that we can confirm if these endophytes are actually consumed as we are consuming the raw vegetables. So one thing that we did quickly was to check the diversity of the endophytes that are contained in these vegetables. And we use different vegetables. Sharon and Raymond, they did a lot of isolation from different vegetables. And that's confirmed. We have endophytes on the vegetables. So, but how do you now prove that they are being consumed? How do you now prove? What's the proof of concept here? So, one experiment that was conducted by Rohan that is here today is the use of irrigation, the spiking of irrigation water. He spiked the irrigation water. Okay, let me backtrack a bit. So here, we ask the question, can human pathogens become endophytic on ready-to-eat vegetables? That's the question we ask. We all know about pathogens. We don't want pathogens in our food. So we ask the question, can human pathogens, can they become endophytic on ready-to-eat vegetables? I would allow you to answer that after this presentation. So we took some pathogens that we know. In this instance, we took Clostridium, and we spiked irrigation water with Clostridium. We took that to the greenhouse. We planted our lettuce, and we irrigated the lettuce. We irrigated the lettuce with the spiked water to see if these endophytes can actually colonize and become endophytic. I'm not going to go into too much details, but 10, 20, 30 days, even up to 40 days, we are still detecting these endophytes in the vegetable. So, and if you harvest your lettuce around that time, definitely you will consume the endophytes that we are talking about as well. So another part of this that we are looking at is the antibiotic resistance. Mariska is working on this, and Moise Akim. They are working on this to check if the antibiotic resistance that are associated with these endophytes that are colonizing the vegetables as well. So here, what we've been able to show is the endophytes, not only the good ones, even because what we should realize here is the endophytes, they are not really pathogenic to plants, but they can be pathogenic to the secondary consumers that will consume the plant. So if you consume the endophyte on the plant, then you may also be consuming, if you consume the vegetable, you may also be consuming pathogenic endophytes sometimes, but not always. So this is the idea that we are talking about. The endophyte moving from the plant onto the leaves, from the rhizosphere in some certain situations onto the leaves, and whether human or mouse can consume them. And when this mouse produces fecal materials, the fecal materials go back into the soil, as well as when it dies, it's also, the endophyte can be released into the soil, and this endophyte can move back into the plants. Yeah, Professor Modise, probably you are thinking, but I'm not a mouse. So I made this video myself to just give an illustration for you to appreciate what I'm talking about in terms of the endophytes. So these are vegetables that are purchased from the store. I wouldn't mention the store because I don't want you to start labeling them or whatever. You can purchase your vegetable in any store. They contain endophytes, I can assure you. And if you are not sure, bring them to my lab. So yeah, these are the vegetables. So we took samples from the vegetables under sterile conditions, and so that's taking samples from the vegetables. So yeah, putting them on the plates. So we surface sterilize them to make sure that we remove all the contaminants that may be on the surface of the plant, because we are looking for the endophytes that are inside the plant. And for us to take them out, we need to give them the nutrient that they need to come out of the plant. So we put them on Petri dishes, and yeah, this is what happens after 24 hours. I don't know if you can see that very well. You can see the plates. What you see on the plate there, you see the endophytes growing out of the leaves. That's your spinach that you consume. So, and it's for all the plants, all of them, they contain endophytes. There is no argument about that. So I hope with that you appreciate the fact that endophytes are on the vegetables. So to wrap up the talk, because of time, this is the typical food chain that we know. We know that the sun shines on the plant, and the grasshopper goes to the plant, it consumes the plant, and the frog is there waiting to consume the grasshopper, and the eagle or the hawk is there trying to consume the frog as well. That's what we know. even little kids in primary school can draw this food chain for you. That's what we know. But are we underestimating the role that endophytes can play in this regard? So I tried to make a video of what can happen in the food chain. So here you have the sun coming out, shining on the plants for the photosynthesis to occur. After photosynthesis, the plants grow, and the grasshopper can consume the endophyte. When the grasshopper consumes the endophyte, if it's the wrong endophyte, if it's pathogenic, the grasshopper will die. So, but if it's not the wrong one, the grasshopper lives, but it lives for it to be consumed by the frog. The frog consumes it. Don't forget, they are all transferring the endophyte from one organism to another. If it is the wrong one that is pathogenic to the frog can also die. But in this case, if the frog doesn't die, it has the endophyte inside it as well, and the eagle can consume the frog. And when it does, it also acquires the endophytes. And when it acquires the endophytes, what happens? It will eventually die anyway, and the endophytes from the hawk or eagle can go into the soil and become endophytic again, and the cycle continues. That's what we are proposing in my research group. So in conclusion, I hope I've been able to convince you that endophytes are important plant microbes that drive different important processes in the ecosystem. Animals are ingesting endophytes every time whenever they consume raw plant materials. That's certain. We are all doing that. Even if you don't eat vegetables, you will eat fruits. It's the same approach. Hence, these endophytes are not only involved in biogeochemical cycling, they are playing very key, important roles in the food chain. So these are the other ongoing projects that, and for my students that are here, I appreciate you coming here. If your project doesn't feature here, it's not because your research is not important. It's because I cannot discuss everything today. So everything that we are doing in the research group is important, but these are other ongoing research topics that we have that are sponsored by different organizations. So these are a few pictures from that I took with my collaborators one time or the other. I like this picture because it was the first time I took a picture with a president of a country. It was the World Food Prize Symposium in Iowa. That was in 2013. Friends, colleagues, there are many friends that are here today. I'm sure you'll be able to see their faces. So they've contributed a lot to my career as well in terms of the support they give me. Always. Colleagues, I don't have the pictures for everyone, but this is just to, this is the last picture I took with my colleagues. This is just to say thank you again for being there. This is the list of the former students. Former and previous students. No, this is not former student. That's a mistake. Sorry. I'm sure my students are worried now that am I disowning them? It's a mistake. So you are still my student. You are still Dr. Kazim. Don't look too worried. We are still together. And thanks for sending those slides last night. You've all worked hard to make this happen. These are the enablers. I call them enablers of whatever that you see here. And I need to also acknowledge the contribution of my past students as well. Most of them are listed here. They've done a lot to contribute to my research. And family and friends that are here, there are many of them, and I appreciate what they've done for me. The support that I've received in my career have been enormous. So I'm very grateful to all of them. Those that are online, that are not able to join us, collaborators, colleagues, friends, families. So thank you very much. Yeah, and the backbone of my support. You know, I always say this to myself. I have many co-authors that I've published with. She is never one of them. But I tell you, she is a co-author of this history that we are writing together. And a single co-author for that matter. So thank you very much, Doyin, for being there for me. I appreciate you. The family, Miriam, Alima, Ibrahim, thank you. Especially, you know, sometimes I would tell you I need to concentrate on what I'm doing. No noise. Thank you for listening. I appreciate that. Yeah, there's nothing like family. I appreciate that. So, and to the sponsors that have always been there for me. Without them, we cannot have the resources to do the research that I've been doing. Yeah, so I want to say thank you to them as well. So, yeah, it's amazing to be here again. I don't want to start thanking people too much because Prof. Balia still needs to say something here. But Prof. Balia, once again, I appreciate you to be here. I mean, that you are here today. We really, the support that I've received from the university, I must acknowledge that, is enormous. We are growing plants every day or whatever. We've got a greenhouse there that we use. The university is really giving us the support we want. Not that I don't want more. Prof. Mbunise knows. So I want more, but I need to, you know, if you say thank you, you can receive more. And I believe I can always receive more. Thank you, everyone, for coming. And this is where I will end this talk today. Thank you.
Occasion here, one of much celebration, to welcome Professor Rasheed Adeleke into the College of Professors at our university. Now, some of you may not know, but this was meant to happen many months ago, but due to unforeseen circumstances, it didn't take place. So it is an extra honor and a reason for much joy that we can have you here today and celebrate this momentous occasion with you. One of the great things about this, friends, ladies and gentlemen, is that it is a celebration of academic achievement. And you heard when Professor Dave Modisi, the Executive Dean of the faculty, read the CV, that to get to this point you had to walk a long road, you had to undertake a journey that took you to many countries of the world and many other universities of this country. In fact, I think you should have rather list of the universities that we didn't associate with, because there would only be a few. The list is so long. And we are honored to have a colleague here today who knows the world, who's been around the block, as it were, the block of science, and who's had all these multiple associations with other universities in the sector, in the country. It brings much prestige and honor to our university, to our campus, and of course to the Faculty of Natural and Agricultural Sciences. Now, nobody should think that we at the university live in ivory towers and that we are divorced from the reality. There is nothing more important and more pertinent today than the issue of food security, which Professor Rasheed has been speaking about. You convinced me today that you cannot be a vegetarian. In the pure sense of the word. When I was a student, I used to work in a fruit and veg store, and we could never get those worms out of those lettuces, because no matter how hard you tried, you would inevitably find some of these moving organisms in the vegetables. So I know that story, and I always wondered what happens to these people, even when they wash the lettuce, they will miss a few, they're going to be eating them. So unwittingly, they will be contradicting their stance on being pure vegetarian. But your lecture today helped me as a layperson in the field to understand a bit more about your discipline and the value that and the difference that it will make in our understanding of food security, the food chain. And it is, when you begin your academic journey as a young person, you always want to make a difference. You want to change the world, and you want to make a lasting contribution. It's very clear that many colleagues in the Faculty of Science, let alone in the discipline that I think you were calling yourself a microbiologist. I'm never sure who's a microbiologist, who's a biochemist, and who is what. I just know they're all in the Faculty of Agriculture and Natural Sciences. But these are the colleagues who are so very necessary, so much in demand, and these are the people who will do, I think, more than many others to save our planet Earth. Who would have imagined that you would be experiencing 6 inches or 10 inches of snow in the month of September on your way to Durban, driving to Durban? Never in the history of this country has that ever happened. It tells you how the world is changing. And so with food supply, we are definitely going to be up to a challenge with our scarce resources, and it is people like you who are going to help us to save this planet. So we really appreciate what you do for the university, for the faculty, but for the world as a whole. May you be blessed, may the rich work that you do, and may you continue to bring new insights, of scientific knowledge, into your discipline, and that will benefit not only your students, but as I have been saying, the world at large. So congratulations to you and your family, and I really want to end by putting emphasis on the fact that when you become a professor, as Professor Rasheed has done and given your inaugural lecture, it is so important for us to remember those who have helped us on this journey. You cannot do it on your own, especially on the continent of Africa, where we struggle against the odds to make a living and to succeed. You had a very strong, family network, supporting community that helped you to move, along the way and achieve your dream. That is so very important. And so today is a celebration of your individual academic achievement. It's a celebration for the university to welcome you to the rank of professor, but it is to acknowledge as well the important role that family plays, the village plays, the people play in making you the person that you have become. Congratulations and welcome.
it only leads me to close this evening. But before I do, we have this is a formal event, but also a celebratory event, and we are now moving into the celebratory event, and that's why we are here tonight. And we'd like to thank Prof. Rasheed for inviting us to celebrate this with you as family, friends, but also colleagues. And as colleagues, I'm sure the faculty members sitting here would like to congratulate you with this work. And at this point, they celebrate with you, the food that you prepared for us, making this a very unique and special event that you put your stamp on. It's very evident if you go outside when we are going to enjoy the food and drink. Endophyte free, I have on So, just final word, we'd like to, as faculty, thank you, Emma and the team, Suzelle, the photographers here tonight for organizing the event. There's a lot of things happening behind the scenes, and thank you again for that, Emma and the team. So, colleagues, please stand up for the national anthem. Thank you.
Così si geleri Africa, manu pagani su ponolayo, Yiswa imitana so yetu, Gosi si gelera, Tina musapolwayo, Murena buluka se chabasa Yesu, ufelice tentuale la sueñe, si buluke, si buluke, si chabasa, Yesu, si chabasa, South Africa, South Africa, Heidi blauw van onze hemel, uit die diepte van ons ziel, voor ons eeuwige gebergtes, wat die krause antwoord geeft. Sounds the call to come together, and united we shall stand. Let us live and strive for freedom in South Africa.
Thank you, ladies and gentlemen. Kindly stand for the academic procession.